Is the screen visible to all of you?
Yes, sir.
Yeah. Okay. Cool. Yes, Arpit, tell me. Sorry, I was occupied. My mind got occupied into figuring out this. What were you saying?
Sir, I was asking about the like we will have any recovery class or something like that.
Yes. So first we'll go with the normal classes and we'll finish as much as I can, as much as we can, and then after that there will be classes like for topics that need a re-revision, a topic that we won't be able to uh, finish within time. So we will have some classes for that. Okay, so don't worry. That's there. We have a reserve. Okay, so I think in the last class we started with the principles and processes, and where did we finish? We finished at plasmid, or artificial recombinant DNA construction technology. Okay, anyway, so anyone would like to give a quick summary? Anyone would like to volunteer for this? Come on, people, you should volunteer now. So I have slight cold, so please bear with my voice. I hope it's clear, right?
Yes.
Yeah. Okay. So if no one is volunteering, how about I go to Samira? Samira, are you there?
Yes, sir.
Would you like to give a quick summary?
Yes, sir.
Yeah. Okay. Great.
So in the previous class, we started a new chapter called biotechnology.
Mhm.
So biotechnology, it deals with techniques of using living system and the components to produce useful products or services for humans. Example, genetic engineering, gene therapy, making RNA or DNA vaccines, and in vitro fertilization. Then we discussed about the principles of biotechnology. So they are mainly two techniques. First one is genetic engineering, and the other is bioprocess engineering. In genetic engineering, we alter the chemical properties of the genetic material. And this modified genetic material can be introduced into the host organism to change the phenotype of that organism. Then in bioprocess engineering, it includes maintaining of a microbial-free environment to facilitate the growth of the desired microbe in large quantity. And bioprocess engineering, it leads to large-scale manufacturing of biotechnological products. Example, enzymes, vaccines, antibiotics, etc.
Correct.
Then to create a recombinant DNA, we use gene cloning and gene transfer. Gene cloning, it is making multiple copies of genes containing a desired, multiple copies of genes containing a desired gene. Then gene transfer is transferring of a gene from one organism to the other organism. Then
what do you say in gene cloning?
it is the transferring of gene from one organism to the other. No, that is gene transfer. Gene cloning is making multiple copies of gene containing a desired gene.
Yes, so it's about making multiple copies. This making word if not clear, then I will write it here. So making multiple identical copies of the gene-sized pieces of DNA. So basically we do it for genes. We can also do it for much smaller or larger piece of DNA. So basically when we are cloning a piece of DNA. Now, if we are cloning the region, which also happens to be a gene, then automatically the term becomes gene cloning. And it's obvious that most of the time we are interested in cloning one or the other gene only, because gene has functions, like especially protein coding genes. They can go on, get translated into the cell to give rise to certain proteins, and those proteins now then will show some effects or function in the cell. So that's why the term gene cloning is there. But basically what we are doing at the fundamental level is making multiple copies of the sequence of our interest. Okay, the DNA sequence of our interest. Is it clear?
Yes, sir.
Yeah. And then what was this origin of replication before we go to transfer? What did we discuss about origin of replication?
The sequence that initiates replication region.
So I have made here the diagram. There are multiple lines coming to an arrow where origin of replication is written. What does that mean? So according to the definition, the region of the DNA that is responsible for initiating the process of replication. Correct. So this is also basically a sequence, a signal sequence from where replication can begin. The replication fork can begin. Remember I told you about the replication fork, chapter 5, when we were studying DNA. Right?
Yes.
Yes.
It opens and then one lagging strand and one leading strand is formed. So it looks like a fork. So two things bifurcate into two more in a semi-conservative manner of DNA replication. So that starts at origin of replication. But what are these multiple points? These are multiple sites of origin of replication. I also told you that there are multiple regions from where replication begins. It never happens that in a DNA, replication starts from one tail end and it goes open the DNA and goes and completes to another end. DNA is very, very big a molecule. So you start replication at multiple sites in the DNA simultaneously also. Okay, so there can be multiple origin of replication in any particular genome. Is it clear, everyone?
Yes, sir.
Okay. Yes, continue, Sumara.
Then we discussed about the plasmid. They are extra chromosomal DNA. They replicate independently of the genetic material.
Okay, so if they are extra chromosomal DNA, how are they stable in the cell? Like, why are they not degraded in the cell? So normally any piece of DNA, when it is present inside the cell, so it starts to degrade. So there are certain molecules about which I will teach you in today's class. They start attacking the ends of this DNA. And also RNA is also not stable due to the similar reason. So why is this stable? Because it's a circular DNA. Okay, it has no loose ends. It's like a circular DNA, which is called a plasmid. And you have studied in a prokaryotic cell. So here I have, this is a prokaryotic cell. It has a genetic material, which is its nucleolus. And apart from that, it has small plasmids. It can have more than one plasmid also, or one plasmid. Mostly it has one plasmid, a single. Sometimes it can be more than one also. So these plasmids are also a piece of DNA, but in a looping circular arrangement, and that's called extra chromosomal DNA. So it's not the part of this one chromosome. So this is, so it's not looking like a chromosome right now, but when it will condense, it will become a chromosome. So every bacteria has just one chromosome because it has just one long thread of DNA, which is its whole genome. Is it clear? And the yes, Samaya, continue about plasmids.
Then we started cloning of our gene of interest.
Before cloning, did you tell about the special property of plasmid? This one?
Yes, they are autosomally replicating circular DNA.
Autonomously.
Autonomously.
Yeah. Autonomous means something which happens on its own and is not under the control of the genome of the cell. So in this case, Autonomously replicating means it can replicate on its like itself without consulting with or without depending on the genetic material of the cell. Yes, then
And we discussed about cloning of our gene of interest. So to clone a specific gene of our interest, a gene sequence, it has to be incorporated with the chromosomes of the host cell, because only the chromosome has the ability to replicate due to the presence of origin of replication.
okay.
Then
But then how does plasmids replicate? If only chromosome has origin of replication, it's not like only chromosomes have origin of replication. Plasmids do replicate, right?
yes, sir.
So you just said above that plasmids can autonomously replicate, which clearly means that plasmids do have their origin of replication, correct?
Yes, sir.
And I also told you that that is how we can construct an artificial recombinant DNA, because we can put our gene of interest, let's say this gene X is our gene of interest, in a plasmid with a marker gene. So we know that if our plasmid is getting replicated and is being expressed, if our gene is being replicated or expressed, then we will also see the marker gene. So this marker gene is antibiotic resistance gene mostly, so that we know that these bacterial colonies, if they are surviving in an antibiotic medium, it means that the plasmid is successfully expressing. And if antibiotic resistance gene is expressing, clearly our gene X, which is also a part of that plasmid, must also be replicating and expressing, correct?
Yes, sir.
And the good thing about plasmids is it can have multiple origin of replication, and also the turnover number, how many times replication will happen, can also be controlled in a plasmid. So you don't have to depend on the division of the cell, of the bacterial cell itself. You can make multiple multiple copies of the plasmid in just one generation of the bacterial division. Yes, continue. So what was this test and control experiment that we did? Samara.
So when we added the antibiotic to the gene of interest, the colony survived. And if we add antibiotic to the empty plasmid, all the colonies died.
Why? Why all died in the empty plasmid and why they
Because
survived?
it didn't have antibiotic resistant gene.
Yes. So because they did not have this plasmid, and in this plasmid they did not have the marker gene. We are checking for the gene of interest, our gene, which we have put, but we also know that it has been put along with a marker gene. So we just have to check for the marker. And if the marker gene is there, then our gene also must be there, correct?
Yes, sir.
And then in the last section we discussed about Stanley Cohen and Herbert Boyer. What did you discuss already? What all have we discussed already?
So only that that they isolated the first antibiotic resistant gene.
Okay, they isolated the first antibiotic resistance gene from the bacteria. Okay, now let's begin from here. So in today's class, I gave you the homework to go and read about endonuclease and exonuclease. Did you do that, everyone? Yes, no?
Yes, sir.
Yeah. Okay, what is endonuclease and what is exonuclease? Let's start from here. What is nuclease, first of all? Tell me. Because both have two terms. The terms are, one is endo and nucleus, right? And then it has exo and nucleus. So what does nuclease means? The
It is a
term.
type of restriction enzyme.
How do you know it's restriction? What do you mean by restriction enzyme? So this is a term given in notebooks, but which part of this word gives you the information that it's a restriction enzyme? So what do you mean by
Sir,
restriction enzyme, Miraj? Your voice is Miraj, your voice is breaking.
Uh,
Yeah. So you introduced a new term, restriction enzyme. You're correct. But what does it mean? What does the word restriction means? Simply. Restriction.
is to hinder something.
To stop something, right?
Yes.
Yeah, okay. Miraj's voice is breaking. Miraj, you would like to come in a good network zone. By that time, let me go to someone else. Okay, Arpit, what does restriction means?
sir, I was answering for the nucleus. Like I was not answering but
Okay.
guessing.
Okay, so let's go there. No problem. We have a lot of things to answer. What does the word nucleus means to you?
Sir, it might be something related to nucleic acid, or it is something made up of nucleic acid.
Yeah, it is something that something has a word called ase in the end. So I've told you what whenever.
Enzyme.
Yes, so that something is enzyme, correct? You figured
Yes,
it
sir.
out. And if it is enzyme and it has nuclei in its as a prefix, let me give you a hint. Amylase, peptidase, lipase. Class 11th, your digestive system chapter. You have already studied nucleases. Nucleases are the enzymes that digest nucleic acids, that digests DNA and RNA, correct? Also, there was an experiment done by Avery McLeod and McCarthy, right? Where they used nucleases, specifically DNases, to see that the Griffith's transforming principle now is not holding up true, correct? And they said that DNA is the genetic material, but no one truly believed them because their experiment was not done in a very clean mode. So people always have doubts. That's why Hershey and Chase has to come up with a brilliant experiment using radioactive sulfur and phosphorus. But you know that nucleases are the enzymes that digests nucleic acids, okay? Now, what does the word endo and exo means? Endo and exo, people, this is simple, right? Endo means
Inside.
inside, and exo means outside. What does restriction means? To restrict or to cut, right? To cut someone away from something. Like if I cut down your social life and do not allow you to meet with your friend, I'm putting a restriction on you, right? Restrictions basically means to not allow the anything to happen. So endonuclease and exonuclease, both of these are enzymes which cut the DNA. Now endonuclease cut the DNA from inside. Like if this is the, I'm making a, let's say this is the DNA, I'm making it straight, non-helical. But you understand these are the bases, right? Let's say this is A, T, A, G, C, C, G, A, T, T, something like that. And towards the end, it is T. Okay, so here also it will be AT, and there will be something towards the end. Here it will be A, let's say. So exonuclease will start cutting the DNA base by base from the sides, okay, from outside. So exo acts from here. So it will first cut this, then keep cutting in this manner or in this manner from this side. But endo is which can cut from anywhere, directly anywhere from the inner regions. Like let's say it can cut CGA directly. So that is endonuclease. Okay. Is it clear, everyone?
Yes, sir.
Exonucleases cleave the DNA from the sides, okay, from the exposed regions. Endonuclease can cut DNA from anywhere in the middle. It just recognize, and both of these work on the principle of sequence recognition. So there are multiple exonucleases and endonucleases, and all of them work on the principle of they recognize certain sequences and they go and cleave the DNA at that particular sequence. So which means there is a specificity in their cutting. It's not like they will cut anywhere randomly. And this specificity is very good. We can now play around with this specificity and use it for our own benefit. I'm so sorry, people. Okay. Now, talking about Stanley Cohen and Herbert Boyer. So they isolated the antibiotic resistance gene. For that, they first have to know that this sequence from here till here is responsible for antibiotic resistance, and it is a gene. So gene you can identify. Let's say you identified a gene in a genome. Now the point is you have to isolate it, you have to cut it out of the genome, right? And to cut, you need something. What do you need? You need these. Yes, tell me. Was it you, Samara? Someone was saying something. No? Okay. So what do we need to cut out a gene? We need these endonuclease, right? Or so endonuclease and exonuclease, they are called restriction enzymes because they cut the DNA. They are also known as molecular scissors. So either restriction enzymes or you call them molecular scissors due to their function. So their function is to just go, and they are molecules. So they go and cut DNA at particular. By cutting, I simply means breaking the bond between nucleotides, and because of that bond breaking, the DNA separates. Now write down restriction enzymes makes the cutting of DNA makes the cutting of DNA at particular specific location in the bracket, write down sequence. Particular specific location and hence are known as molecular scissors. And hence are known as molecular scissors. So how did they figure out? How did they Stanley Cohen and Herbert Boyer isolated after recognizing that this gene, this region, let's say a long DNA was there, and they realized that from this part to this part is the region that gives antibiotic resistance. So if they can just cut from here and here, and the next part is, so they have isolated it from any one kind of bacteria that is resistance. Now you want to express this in any bacteria of your choice. How can you do that? After telling all of everything that I have told till now, anyone would like to give me an experimental plan? I figured out an antibiotic resistance gene in a bacteria. I identified it, first step. Using restriction enzymes, which is endonuclease, I cut it out, second step. Now, what? I want to use it as a marker for my experiments to make the recombinant DNA technology. So what will I do?
We can transfer the gene from that to the other, our host vector bacteria. By using lipase, we can attach it to the plasmid.
We can attach it to the plasmid. Yes, that's the third step. Before transferring, not lipase, it's ligase. Okay, lipase is the enzyme that degrades lipids. Okay, did you say ligase or lipase?
Lipase, I'm sorry.
Yeah, it's ligase, Lig. Okay, so after cutting that part using endonuclease, you need to put it in a vector that can take it to the next bacteria, or any bacteria that does not have it naturally, but you can give it naturally. So what, write down the cut piece of DNA. The cut piece of DNA which in this case, what is that cut piece? Antibiotic resistance gene. It can be anything, right? Now you must be wondering that how does these restriction enzymes, they're called restriction endonucleases, how do they work? So how do they work is they only let's say identify this, let's say ACC-AGG, this sequence of 6 base pairs coming together. Now what is the probability of 6 base pairs coming together all the time? You must be thinking that it's very high, right? What are the chances that ACC-AGG will be repeated?
So it's very,
Sorry, yes, Miraj?
so it is very low, the chance of them coming.
It's very low, right? But
So
if I say what is the chance of ACC coming together? It is lower or higher than this? So this is first case, this is second case. Whose chance is higher of repeating?
there is
ACC, right? The second case, correct? If I say AT, repeating this, even higher, right? Or if I go something more than that, right? If I say ACC, AG, CT, then the chances are even less. So the bigger the fragment, the lower are the chances that it will repeat. You know, any fragment which is around, let's say 20 base pair long, and it's specific, the chances that it will randomly repeat in the genome is so low that it will repeat after so many base pairs that it will be, the number will be higher than the whole genome. Okay, so that's why in this case, for these restriction endonuclease enzymes, they mostly identify and cut either at 4 bases, a sequence of 4 base that repeats, or a 6 base that repeats, sometimes 3 also. but 4 and 6. So 4 can come, okay, but at whenever it comes, we know where it is in the genome, because we can do genome sequencing for any organism, and we can just, once the genome sequence result has come, we can put it in the computer, in your Word or text or anywhere, just go to search and type that. You must have been doing it in Word, right? Control plus F, then you put your name. Suppose there is a paragraph in which you want to highlight all the words which are your name. Let's say Samira wants to figure out where is Samira written in that whole document, how many times. So you do Control plus F, go and write Samira. It will tell you where all Samira is, right? Yes or no? Similarly, there can be a genome sequence file. You can just search where that whatever site. So there are many endonucleases. Some famous one are EcoR1, Hind3, Hind2, etc., etc. So you know that for this endonuclease, this is the site which it cleaves at. You will see what all places in that DNA sequence that site is present, how many times it is present. And if it suits your experiment or your interest, then you use that restriction endonuclease to cleave your DNA. In this case, because you know the sites, you will also know the fragment of your DNA. You will know that how big or small your fragment will be after the restriction endonuclease has cleaved. Correct? Everyone understood?
Yes, sir.
Yes, sir.
So the cut piece of the DNA, which in this case is antibiotic resistance gene, is then ligated, or you can say, right, attached as well, ligated to the plasmid. And the enzyme that helps in this, ligase. Remember DNA ligase in DNA replication? When Okazaki fragments were being created in a lagging strand, right? In semi-conservative method of DNA replication, one strand was leading, a continuous strand, the other strand was lagging strand, correct? So it will make small fragments. Remember, people, yes or no?
Yes, sir.
Those small fragments were called Okazaki fragments. And how does those small fragments were then put together to make the whole DNA?
By DNA ligase.
By the enzyme DNA ligase. So the same enzyme we are using here, just like restriction endonucleases, there are ligases also, multiple kinds of ligases that stick different sequence or different bases of in the DNA. Now, once you have stitched or linked it or attached it or ligated it, whatever word you want to use, with your plasmid DNA, now your plasmid DNA will start acting as a vector, right? Vector for gene transfer. So do you see what are we doing here? If you pay attention, we are taking all the raw materials from the nature. We realized that, okay, first we knew that bacteria have an extra chromosomal DNA in the form of plasmids. Bacteria do their normal routine things using their plasmid. We realized, okay, what if I isolate those plasmids first? We did that. Then we realized it is possible to cut DNA at specific locations using restriction endonuclease. We did that. Then we realized whatever piece of DNA we are cutting, if we put it in the plasmid and then put that plasmid in the bacteria, the plasmid can act as our own vector, where we can make any kind of DNA, we can make any recombinant DNA that we want to be expressed in a bacterial cell, correct? So we are just using living systems and their products to make technology or technique for giving any service or product to humanity. Now we are just talking about the mechanism here. We have not reached to what are the services that we can do with this. But you realize still here that it is a vector for gene transfer. Plasmid is a vector for gene transfer. Now, I already told you, so this is a plasmid. I already told you that along with antibiotic resistance gene, because we don't want to give bacteria antibiotic resistance. You know, what is our benefit in that? It is bacteria's benefit for sure. But what is our benefit? No benefit, right? And we will never give anything to a bacteria where it is not beneficial to us and only beneficial to bacteria. So we're very selfish as species. So we will also like to attach something along with antibiotic resistance gene, and that will be any gene that we want to express. It could be any any enzyme that we need, any other molecule or protein that we need to be synthesized, and then we purify it and sell it for like to humans for their benefit, therapy, treatment, cure as a medicine, etc., etc. So, and we also want that whichever bacteria which is having our gene of interest should survive in antibiotic conditions, because that is the testing condition where we will test those who do not have our gene and do not have the plasmid that we have created, hence do not have antibiotic resistance, will die. Till now, everything clear? How to make a recombinant DNA, right?
Yes.
But one question is, why will a bacteria take up the plasmid that we have created? It can just say, no, I don't want your plasmid. I have my own plasmid. Or I don't have a plasmid, I don't need a plasmid. Thank you so much. You know, I'm good. How to put it, how to be sure that we will end up putting that plasmid? What if we develop this whole technology and then we realize, oh, we cannot put it in the bacteria. Bacteria is not taking it up. Correct? So how to make sure that we will always give it to the bacteria? Anything? That's why I ask you in the very first, this was your another homework. I hope you did it. What are competent cells? Anyone who have figured out what are competent cells? Anyone? What is a competent bacteria cell?
They are the bacterial cells that take up the foreign DNA from the surrounding.
Yeah, so what do we, how do we, can we make any bacterial cell competent? So you are perfectly correct. So Sumaira said that competent bacterial cells are those bacterial cells which can easily take up any foreign DNA present in the surrounding. Correct. But are all bacteria always competent? No. Can we make a bacteria competent? Or any cell competent for that matter? Yes, we can. That's called making competent cells. So how can we do that is, to make any bacteria competent, we just need to make the bacteria open.
They're like gel electrophoresis.
Gel
Gel
electrophoresis
plas-
is, yeah, continue. Complete first.
Sir, in Argos gel electrophoresis,
Agros.
here we give shock. Ma'am, sir, we first heat the bacteria and then cool it to inject a host phenotype into the bacterial cell.
Yes. So this host phenotype, you are are you reading from somewhere? Are
Oh,
you
sir, I studied the chapter.
okay? But agarose gel is like it already had pores. Okay. So in agarose gel, so you can make different concentration of agarose gel, and depending on that concentration of how much agarose is there, 1%, 2%, 0.5%, 3%, the pore size of that gel will be decided. Now agarose gel is used to uh, differentiate between bigger molecules and smaller molecules. Okay, so on a gel, smaller molecules run faster and bigger molecules run slower, because clearly if it is a pore size, if I ask 10 people from age 5 to age 35 to pass through a small hole, so a kid who can easily crawl through will first clear, right? Others will struggle. The biggest 35 years old might be just stuck there at the beginning, correct? So that's the idea. And how do we push? The push is the electromotive, the voltage dependent push, because DNA is DNA is negatively charged. So if you put an electrode and run a voltage, DNA will be pushed from a negative terminal to a positive terminal. It will be attracted. So you are pushing or pulling, and also you are making the DNA to pass through different size of pores. So the smallest piece of DNA will move the fastest. That's a different technique. But you're correct that what we have to do is we have to make pores in a bacteria. Now, how to make pores? So to let you know, there are already very small, minute pores or channels present in a bacterial cell wall and membrane. We just have to increase its size for a moment, instantaneously or momentarily, during which our plasmid can just pass through that pore and get into the bacteria. So that we do through shock. And he's completely right that we can do it through a heat shock. So what you do is, it's called, you keep a bacteria on ice, cool it, okay? Then suddenly ice is around 0 degrees Celsius. So from that ice, we just put it back on 42. Now 42 from ice, 42 or 35 or 40 even, it's not a temperature at which bacteria will die. So we don't have to kill the bacteria. If they get killed due to heat, the whole process is gone. So we don't have to kill it. You just have to heat it, then cool it, and then again heat it. So cool, heat, then cool, and then heat. It's called heat shock. And due to heat shock, the pores kind of open and the plasmid enters. Just that we need, okay? Or you can do it using divalent cations. You know what is a cation? Divalent cation. Cation is a positively charged atom. Anion is a negatively charged atom. You know these things or not, people?
Yes, sir.
Yes,
Yeah.
sir.
And what do I mean by divalent cation?
With the valency too.
I'm so sorry. Yes, the valency has to be 2. So can you give me an example of a divalent cation, people?
Cu2 plus.
CO2. CO2 plus? What is CO2 plus? CO2 is carbon dioxide. All the valencies here are satisfied.
Cu2 plus.
Cu, you mean copper?
Yes.
Ah, okay. You can say copper. So Cu2 plus, is it correct? Is copper divalent? People? Anything else? Yes, no, say something. Can I use calcium also? Yes, no. Anything will work. Yep. So exactly we do that. So we use calcium ions in making cells competent. Okay. Heat shock and then bombarding it with calcium ions. So what calcium ion done is these are big ions. So when the moment you open through a heat shock, you make the pores of a bacteria open and become big. These divalent cations help in the process and also help the plasmid to go inside, okay, the bacterial cell. So you can write down one line. We can transfer the plasmid containing our gene of interest, or let's write it like this. In order to transfer a plasmid, containing our gene of interest in a bacteria,
Plasmid.
containing our gene of interest in a bacteria, first we need to make the bacterial cells competent. First, we need to make the bacterial cells competent. Now let's define competent bacterial cells. Write down. A competent bacterial cell can take up foreign DNA or plasmid from its surrounding. Inside the cytoplasm. Okay. So that's how we make sure that all this process does not go waste and we successfully end up putting our gene of interest or our recombinant DNA into the cells. Okay. So till now, just to recap what we have studied, there are 3 steps that we have to follow whenever we want to genetically modify any organism. First, we have to identify the gene or the DNA that have the desirable or desired characters that we want to transfer. So first is identification of our gene. Second is we have to introduce that DNA fragment into the host in whichever cell we want to express it. And third is when we have introduced, we have to maintain that introduced DNA so that it stays stable. For that matter, I can inject any DNA into a cell. We don't have to go through all of this process. I can just inject it into a cell, but it will not be stable there. DNA gets degraded by enzymes called nucleases. So we have to maintain it inside the host so that the host can transfer it to its progenies. Is it clear, everyone?
Yes, sir.
Okay. So this part is clear. Now let's study about some REs, restriction endonuclease, in detail, because these are the bread and butter, okay, for biotechnologists. So any genetic engineer, any engineer the tools that an engineer used to cut a metal, put it, mold it. So you will see a civil engineer using saw to cut things, and then using what do they use to join metals? What is it called? Welding machines, right? So engineering is all about cutting, joining, yes or no, and making something new. You understand why we call it bioengineering? We are doing the same things that engineers, a mechanical or a civil engineer does on a macro level. Cuts, join things, make new structures, make new engines, put things together to make something functional. We are doing the same thing here. So our tools, for a bioengineer, the tools that cut are REs. Now you must have some of the important ones are Hind2. Okay, Hind2. Okay. Now, before I go forward, let me tell you, there is a way by which we name these restriction endonucleases. Anyone knows about it? So what we are going to read about in this part is eco RI and Hind2. We'll focus on these two restriction endonucleases. Okay, now let's take an example. So let's start with, you have heard of E. coli, right? Everyone?
Yes, sir.
So eco RI or R1, it's actually Roman 1. So in this name, this is the name of a restriction endonuclease. So it's clear that that the word eco and R comes from eco comes from
Equal.
yes. What is E. coli? Full form? Escherichia coli. If you don't know, you should know it. Escherichia, ESCHERICHIN. Escherichia coli. It is the name of the bacteria from which this new endonuclease has been derived from or taken out from. Now this R tells you about the strain, R strain of E. coli. Okay, and this one tells you the order of, you can say, discovery or isolation. Okay, so if it is eco R2, what does it mean? What does it mean? It means that restriction endonuclease is derived from E. coli. The strain is again the same strain, but it came after one. So that's the second endonuclease that has come from E. coli bacteria. So first, always tells you about the name of the organism from which that restriction endonuclease has been derived from. Second talks about the strain, and then the Roman numerical talks about the order of isolation. Is it clear, everyone?
Yes, sir.
So anyone knows where does Hind2 comes from? Okay, anyone would like to do a quick search right now? Tell me. You all have internet with you. So you know one thing, that 2 means it is the second one that has been isolated from the same. So what does H stands for? What does HIND stands for? Quickly, I'm waiting, people.
Haemophilus influenza.
Yes, perfect. Very good. HIND2. Okay, so tell us. HIN stands for?
Haemophilus influenza.
And you have studied this before? You are already familiar with this bacteria. What does it do? What does Haemophilus influenza do? It causes pneumonia. Remember, people? Streptococcus pneumonia and Haemophilus influenza both causes pneumonia, the disease. What does D stands for? Clearly the strain of this bacteria, right? What is D? Have you figured out the strain? Okay, so in this case, I forgot to tell you. The strain is RY13. RY13 strain of E. coli. What is have they given the strain here? Who was answering?
The RD strain.
Which strain?
RD.
RD. So D is from here. Okay. RD strain. And then you know what does 2 means? It is second enzyme to be isolated from the same strain of the same bacteria. Everyone is clear with these namings of restriction endonuclease?
Yes, sir.
Yeah. Now, just to tell you, scientists still now have figured out somewhere close to 1,000 restriction enzymes, and I'm not updated. I think even more than that, we know. I think in your NCRT, they talk about a lesser number. So, but they're somewhere around 900 to 1,000 REs, restriction endonucleases, restriction enzymes, both including endo and exo, that we have isolated, like scientists have isolated from more than 250 strains. More than 250 bacterial strains. Okay, so there are many restriction endonucleases, and the beauty is that all having different and unique, all having different or unique recognition sites, recognition site sequence. And these site sequences are small or big. You know, it can be a 3 bases, 4 bases, 6 bases, in some cases 8 bases or more also. So if whatever experiment or research you are carrying, and whatever fragment you want to make, wherever you want to cut your DNA, if you know that there is a restriction enzyme available, you will use that. Okay. Okay, so let's say there are 2 restriction enzymes, EcoR1 and HIN2. You want to use EcoR1, but the site that EcoR1 recognizes is present at the 2 ends of your gene of interest, but is also present in the center of your gene of interest, let's say. So will you use EcoRI to make a recombinant DNA for your gene of interest? Will you use that?
Can you say again?
Yeah. So let's say you want to use EcoR1 for your research to basically to cut your gene out. So it has the recognition site of EcoR1 is present before your gene starts and after your gene ends, but also it is present in somewhere in the middle of the gene, that site. So will you use EcoR1 for your research or not? Anyone? Do you want me to repeat again, people? If it's not clear, you can. I will repeat. Let me draw it for you. So we want, this is our gene of interest in a long piece of DNA. So what is marked with green is the gene that I'm interested in. So I want to cut this gene out, make a plasmid of it, or put it in a plasmid, then put it in a bacterial cell and make multiple copies of this gene. I want to do gene cloning for this gene. Okay. Now I want to use EcoR1 to cut it out. It's the restriction endonuclease. And EcoR1 has a recognition site here, has a recognition site here, and has a recognition site here. And this recognition site, let's say, for example, is C. Let's say it's 4 base pair long. So the 4 base pair are present here also, and here also. So will I use this? Should I use this or not? Or let me do multiple. Then I'm using 9-2. Let's say, for example, this also has 4. Okay, base pair is not important, so I'm not telling you about base pair. But I'll tell you where the sites are. For hind 2, the site is here and here. And then I'm using one more enzyme, let's say RE alpha, let's say. It is an imaginary restriction enzyme that I have taken, and it has sites here and here. Okay, now you have to cut your gene of interest, which is this piece, to make a plasmid. Which restriction enzyme will you use and why? So the question is for Alia. You're there, Alia? Yes.
I think.
Sorry?
I think it should be hint 2. I'm not very sure, but if we take eco R1,
Okay.
it would be there will be unnecessary segments too. I think because maybe I'm not very sure.
There will be unnecessarily.
Segments like this.
So you are
Yeah.
saying that this fragment here is unnecessary and here also it's unnecessary, right? But don't you think that's also with your hint, because
Yeah.
yeah,
I'm not very
so
sure.
you will biology is never perfect. Okay, Alia, your answer is really good and amazing. Your thought process is very rational. So we would, one criteria that we always look for when we are choosing restriction enzymes for our experiments or for to do biotechnological things is that we should not get lots and lots of flanking region, which is not our gene. Why should we carry a burden of thing which is not important to us? So we want to get it as close to our gene as we can, but it's never going to happen, right? I should not say never, but negligible chance that you will not get flanking region. You will get flanking region, because how can a restriction enzyme know that this is your gene of interest, so I should cut from here and cut from here and make your life very easy as a scientist? No, our life is very difficult. So we have to bear with some flanking regions, of course, like all the time. And you're right. So you would like to go with Hin2 because, but the reason that you gave also applies to Hin2. Even in fact, if I see Hind2 here has a bigger flanking region than Eco R1. And on this side, Eco R1 has a bigger flanking region than Hin2. Correct, Alia?
Yes, sir.
So anything else you would like to? Or you can also say that we can use any of the two. Can we use both Eco R1 and Hin2?
Yeah, I think maybe.
Okay, so Alia is also up for Eco R1. What about RE alpha? This is one.
No, because because it cuts between like that the DNA segment we made it's like it's from there. Yeah, so it's not.
Yeah, it's cutting this part, right? We don't need this. Correct? So clearly this is a cross. Everyone agrees that we are not going to use RE alpha?
Yes, sir.
Yes, sir.
Now the choice is between Eco R1 and Hin2. So Alia thinks that, okay, we can go for both because in one we have to, compromise at this flank, and in the other we have to compromise in this flank. Anyone who would like to, who thinks otherwise? Or everyone agrees we can use both? What about you, Myra? And Reem, you're not speaking today. What do you think, Reem? Reem, Wafia, Samiha? Would you like to go with your batchmate, Alia? And agree that we can use any of the two? So Myra, what do you think? Anyone, just one person who would like to give some inputs, then I'll tell you the right answer. Okay, no one wants to give any inputs. Fine. So the right answer is we can only use Hind2 in this case. We can also not use Eco R1 if we want to get this gene fragment, because Eco R1 has this 1, 2, and 3 sites. So it will cut here, here, and here. It cannot differentiate. It will not. So the fragments that we will get is this one, and this one. This is also not our gene. This is also not our gene because it is cutting from half, and this is also not our gene. You understand, everyone? The problem with Eco R1.
Yes, sir.
So, and restriction enzymes know nothing. They just know whenever they see their recognition sequence, they will go and cut the DNA at that point. All they need to know is that recognition sequence. If the recognition sequence comes anywhere inside our gene, then we cannot use that restriction enzyme for our experiments, or for that experiment where we want to clone that gene. So the only option we have is Hin3, because it will cut here and here, and this whole fragment, though it has some flanking region, so this part and this part is waste, but it has our gene also, which is intact. Correct? So we can easily clone it inside a bacteria by attaching this piece to a plasmid and then sending it into the bacteria by making it a competent cell. Everyone understood about REs?
Yes,
Yes,
sir.
sir.
Yeah. And how to select REs for your experiment, that is also clear.
Yes, sir.
Yes.
So you will not get confused, right? If you see the sequence anywhere, recognition sequence anywhere in the middle. Now, we can also use it in one another way. Suppose you want to like make fragments of a gene. You want to make fragments of the gene itself. I don't know in what kind of experiment would you like to do that, but let's say we want to make fragments of the gene and not clone it. We don't want to take it intact. We want to break it. Then we can use echo R1 here, right? If we want to cut the gene, right? The gene becomes, it's non-functional. Correct? But mostly we do it for cloning. So mostly we will use the REs, which can give us a clean segment containing our gene of interest. So write down, we select the REs, we select the restriction enzymes, which we select those restriction enzymes for gene cloning. We select those restriction enzymes for gene cloning, which give us a clean DNA fragment, a single clean DNA fragment containing our gene of interest. Okay. Now write down next point, restriction enzymes.
So can you say what's after clean?
Sorry. Can anyone just say a few words before and after? Sumaira?
We select those restriction enzyme for gene cloning, which give us a single
Yes, which
DNA.
gives us a single and clean DNA fragment containing our gene of interest. Okay. Containing our gene of interest. Yes. Now, restriction enzymes are of two types, as I told you, exonuclease and endonuclease. Just write down a few words about these as well, if it is asked. It can be asked in your school exams or something. So write down exonuclease remove the nucleotides or cleave the nucleotides. Remove is a simple word. Exonuclease, exonuclease remove the nucleotides from the ends of the DNA fragment. From the ends, two ends, okay? From the ends of the DNA fragments. Or in other words, you can write, they degrade the DNA outside in. So they start degrading from the outside, from the two ends, and then they keep degrading. They cut the DNA from outside in an outside in manner. I'm sorry, I'm making too many changes, but yeah, you understood, right, everyone?
Yes, sir.
Whereas endonuclease, write down, endonuclease makes the cut at specific sequence positions within the DNA. Specific sequence positions within the DNA.
At
Specific sequence position within the DNA. Anywhere within, not from the side, anywhere within the DNA. Now, what are these sequence that I'm saying that they recognize, right? I told you that they can be of varied length, sometimes 4, 6, 8, any number of base pairs that they will recognize. But write down the recognition sites. The recognition sites are palindromes. You know what are palindromes? Anyone know what is a palindrome? A palindrome is a word which spells the same when you spell it forward and backward. Just to give you an example, the name Nitin is a palindrome, because if you spell it from here, it's Nitin, but if you spell it from here, it's Nitin. Do you understand?
Yes, sir.
Yeah, so these are palindromes. So if a sequence is palindromic, it does not matter where you read it from, it will read the same. Okay, that's how endonuclease or exonucleases, when they recognize their sequences, especially endonucleases, they work all the time. Another example is, I think the word Malayalam is also a palindrome, right? Because I remember it. I
Yes,
studied
sir.
it in NCERT. I don't know if it's there or not. The word Malayalam is an let's write it. Malayalam, right? Malayalam. Yeah. If you put this A here, then only it's a palindrome. Some people write Malayalam without this A. So it's Malayalam. Malayalam. Yes. Many words in our day-to-day life are palindromes. Okay. So these recognition sites are also palindromes. Can you give me? So let's make a palindromic sequence. Anyone? Can you come up with any sequence that is a palindrome? Let's keep it 6. Just 6 base pairs. So that the 5' to 3' reads the same as 3' to 5'. So let's say can I write anyone? Simple, you just have to make it the same. So whatever is here has to be, so whatever you read here, let's say A, so it has to be A here. Now you make T, so it has to be T here. You make G, so it has to be G here. Now, because you are making a 6 base pair long sequence, now you just go with the pairing. What binds with G? C, right? What binds with T?
A,
A. What binds with A?
T.
Yes. So if you read it, so this sequence is a palindromic sequence. ATGCAT. ATGCAT. Correct? Do you see this is a palindrome that we made? So
Yes,
this could be a recognition site for any endonuclease. Maybe this could be a real recognition site. We just made it in the class. But you understand where do they go and bind? So their recognition sites are, and then they cut somewhere inside this. And when they cut, they always leave the sticky ends. Okay. So restriction enzyme do not cut clean. So what if they cut like this? It's a clean cut, right? If you leave a clean cut, then point is you are not leaving sticky end, because one of the benefit of using restriction enzymes is they leave a sticky end so that you can stick the cut sequence somewhere else. So how do they cut is they cut like this. Let's say they are cutting here. So they will cut here. Okay. So now you will be left with AC here, which can go and bind with TG. Okay. Does it make sense, everyone? So always cut like blunt, like this or this or this, so that you leave some sticky ends. That restriction enzymes do. So write down restriction enzymes cut the DNA strand away from the center of the palindromic sequence. Away from the center. From the palindromic sequence. But between the same base pairs on both.
Could you repeat the question again?
It's not a question.
Sorry,
It's like,
not the question, the
I'm saying
statement.
that the whole idea is that restriction endonucleases, when they cut, if they make a clean cut, let's say if they cut here, so this fragment goes this side and this fragment goes this side, this side. So it's not very useful to us, right? Because we just don't want to cut and take it out. We also have to ligase it somewhere. We have to attach it into the plasmid to make it a recombinant DNA. So the benefit with recombinant restriction enzymes is they cut in a manner which is away from the center and which leaves sticky ends. So how they cut is, let's say if they are cutting between C and A here, so they will cut between C and A here, between the same base pairs on two strands. So if you see, this is how they cut. Okay. So when they do this, what was here? C, here was G. Yes. So when they do this, as a result, what is the result that you get? You get ATGC. G and C will be unbound. And here it will be TA. So you will get this, or the next other fragment is ATGC, right? So you get these sticky ends, right? Which you can put. So if you see the structure, have you played that game? It's a mobile game where different shapes fall from the top, right? And you have to fit them all in when they come to the base, right? So let's say if a shape like this has fallen, what will be the next shape you would like? Something which is like this, right? You have not played this game? These keeps coming down like this. I'm talking about my time. People, yes or no? Do you understand? So these two fit well because they have their ends here. You know, they have these sticky ends here which fit into each other. So if you look at when a restriction endonucleases cut, they cut in this shape. Can you see? Now here is this one sticky end, which we can use to ligate with something. Do you understand, people? So it's a diagonal cut. It's not a clean cut. So that's what I'm dictating, that restriction enzymes cut the DNA strands away from the center of the palindrome, away from the center of the palindromic sequence. So this is the center. They are not cutting from here. They're cutting a little away from the center of the palindromic sequence, but between the same two bases. But between the same two bases on the opposite strands. But between the same two bases on the opposite strands. Now this makes sticky ends on each strand. This makes sticky ends on each strand and helps the action of the enzyme DNA ligase and helps the action of the enzyme DNA ligase to join the sequences back or join the sequences with something else. Is it clear, everyone?
Yes, sir.
Yep. Okay. So the whole recombinant DNA technology is clear to you.
Yes, sir.
Very good. So write down a homework that you have to do today before you come tomorrow. Tomorrow I'll go into some applications of this and we'll talk about some vectors. But today you have to read. If you have NCERT, please come to page 197 of your book. You will see there is a diagram, a diagrammatic representation. Can you see, everyone?
Yes, sir.
Okay. And also page 198. So you have to read these two pages. These two pages talk about this whole scheme. And then what I think, if I'm not wrong, Saifullah was talking about gel electrophoresis, right? That is where when we cut the DNA, how can we now separate them on the basis of their sizes? Okay. So these two things you have to read on your own. Okay. And if you have any doubts, I will take the doubts in the next class. And then I'll start with some plasmids. So we have made plasmid till here. The plasmid is clear. Now we have to just read about plasmids. They are called vectors, or also known as cloning vectors, because we are making them artificially. So they are called cloning vectors. BSE, YSE, we have studied about when we were studying human genome fingerprinting and human genome project. I told you about two vectors, bacterial artificial chromosome and yeast artificial chromosome, BSE and YSE, BAC and YAC. We'll talk about it also. And then we will go for PCR in the next class. Okay. Anyone has any doubts which you want to ask? I'll stop here for questions. I'm so sorry. I'm not completely well today, so please bear with me.